Hello, I'm Annemieke Aartsma-Rus from the Leidnu University Medical Center, and I was asked to present on therapies for Duchenne that are approved or in development. Sadly, I cannot be with you in person because I had to be at another conference, but instead I'm recording this video and I hope it will be useful for you. So before going into the content, I will give my disclosure. So the executive summary is that my employer, the Leiden University Medical Center, has patents on exon skipping, which is one of the topics I will talk about today. And as an inventor, I'm entitled to a share of royalty. And also, I will present on different therapeutic approaches and some of the pharmaceutical companies that develop these approaches. I have been a consulting, or I still am a consulting, and I have been or am in advisory boards. And all the remuneration for that goes to the LUMC, so my employer, and not to me personally. So with that out of the way, what are we going to talk today about? I will briefly explain the context. So what is Duchenne and how does dystrophin lead to pathology? I will introduce the different steps of therapy development, and then I'll focus on therapies that are approved or in late-stage clinical development and explain how they work, what they can achieve and what they cannot achieve, so what to expect from a therapeutic angle, and what evidence is there currently available for patients to suggest that these treatments work, and which outstanding questions are there still. And I will do this both for dystrophin restoring therapies and for therapies targeting pathology. So as said, first some context. So Duchenne patients have Duchenne because they don't have the dystrophin protein. And the dystrophin protein normally stabilizes muscle fibers during contraction. And dystrophin does this by connecting the skeleton of muscle fibers that ensures that the muscle fibers contract to the connective tissue surrounding each muscle fiber. And in that way, it resembles a carabiner, which, like dystrophin, has two domains that are connected to something else. And like the carabiner, you don't need the carabiner to go rock climbing. You need it to prevent things going wrong. And like that, you don't need dystrophin to have muscles. You don't need dystrophin to use your muscles. You need dystrophin to prevent things going wrong. And if there's no dystrophin, like in Duchenne, you have continuous muscle damage. And this will then lead to chronic inflammation, impaired regeneration, the formation of scar tissue. So instead of muscle, there's fibrosis and adipose tissues. And with time, the loss of muscle tissue will also result in the loss of muscle function. Duchenne patients make no functional dystrophin, while Becker patients make partially functional dystrophin. And I'll explain why these two dystrophins are formed in Duchenne and Becker shortly. But it's good to bear in mind that in both diseases, these processes happen. So there's there's chronic inflammation, impaired regeneration, and scar tissue formation. It's just that in Becker patients, these processes happen slower. So the genetic explanation for why both Duchenne and Becker are caused by mutations in the dystrophin gene, first we need to understand how the dystrophin gene acts normally. So this is the genetic code for the dystrophin protein, and you can see it's dispersed over the gene in 79 of these puzzle pieces, and we call these exons. So the exons contain the gene code for the protein. Now, before the gene can be translated into protein, several steps have to happen. So we have the gene, and first the gene makes a temporary copy of RNA. And this temporary copy contains these exons, so the coding pieces, but also the pieces in between that do not contain protein information, the introns. And before this gene code can be translated, the non-coding pieces have to be removed during a process called splicing. And after splicing, we end up with only the exon, so only the genetic code. And this is then translated into protein. And in case of the dystrophin gene and the dystrophin messenger RNA, this is then translated by the cell into the dystrophin protein. And this is a process that's extremely complex, but it happens in all our cells. And fortunately, we don't have to think about it. Our cells know what to do. Now, what happens in Duchenne patients is that usually one or more exons are missing. And you see here an example, exon 48, 49, and 50 are missing. And you see also 47 doesn't fit to 51. So we then say the reading frame is disrupted. So the genetic code becomes unreadable. And what happens is that the cell will translate the code into dystrophin, But here in exon 51, the code becomes unreadable. So it can only translate the beginning of the protein and then translation stops. And this means you get only the beginning of the protein that lacks that second domain to connect to the extracellular matrix, to the connective tissue surrounding the muscle fiber. And therefore, these dystrophins are not functional. And these are the dystrophins that are found in Duchenne patients. What happens in Becker patients is that also one or more of these pieces are missing. So we see here 48, 49, 50, but now also 51 is missing. And we also see that 47 fits to 52. So now we say the reading frame is not disrupted. The code maintains readable. So the cell can translate this from start from 1 to 79. It's just that in the middle, there's a piece missing. Now, because the crucial domains for dystrophin function are located at the beginning and the end, these dystrophins are partially functional. They're slightly shorter because part of the code is missing, but the crucial domains for function are there. And this is why Becker patients have these partially functional dystrophins, have a later onset of symptoms and a slower disease progression. So we're back to our carabiner example. I hope you now understand why Duchenne patients make only the beginning of the protein, while Becker patients make a protein that contains both crucial domains. So when we are developing therapies, there are some challenges when the disease is a muscle disease. The first challenge is that muscle is very abundant. So 30 to 40% of our body weight is muscle. And it's also not one big muscle. We have over 700 different muscles. And almost all are affected in Duchenne. So if you want the therapy to work, you need to deliver it to not just one muscle fiber in one muscle, but to the majority of muscle fibers in all affected muscles. And sadly, once muscle is lost, this loss is irreversible. So the fat and fibrosis, once that is there, this will not return to muscle. And that means the time of intervention matters. So I'm not saying that once someone loses ambulation, there's no point in treating. When someone loses ambulation, you can still maintain respiratory function, you can maintain arm function, so there's definitely merit in slowing down disease progression. However, it's not realistic to think that when someone loses ambulation, that you can treat them and that they will regain ambulation. Once that function is lost, it is irreversibly lost, sadly. So now something about the different steps of therapy development, just that you understand how it works and have a realistic expectation. So when we have an idea as researchers, usually what we first do, we test in cultured cells because cultured cells are easy. It's a 2D layer of muscle cells, and we have Duchenne muscle cells, and then we can treat them. So we have our therapeutic idea, we can treat the cells, and it's easy because it's a 2D layer, we can just add it directly. We don't have to worry about whether the bloodstream will reach the cells, whether there's an immune system that is triggered, because there is no immune system in these cell cultures. There are no organs, so there's no liver breaking down the treatment, there's no kidney clearing the treatment. So it's very simple. But when it works, then we can move to the more difficult models. And usually these are animal models. And for Duchenne, usually these are mice. So we have mice that don't make mouse dystrophin and therefore also have dystrophic pathology. And of course, these mice have a bloodstream, a circulation, organs, and immunity. So it's closer to the human situation. And therefore, it's also more difficult to use this. But when we show that something works in cells and in mice, then we can move to patients. But then we have to bear in mind several limitations. And the first one is that the mouse regenerates really well. So while the mouse also has dystrophic pathology, the muscle quality of the mouse is much better than in patients. So this means that we will always overestimate the therapeutic effect that we see in the mouse, because it will always be better than what we can do in patients, because the muscle quality in patients will be less. And secondly, a mouse has a really high metabolism, which means that what is safe in the mouse is not necessarily safe in the human. And that is why if we move to the clinical trial phase, we try very carefully. We start first to see whether the treatment is safe and how the body responds to the treatment. And then when we have this information, we move to the later stage trial, like the phase 2 and phase 3 trial with control groups, to see whether the treatment is also effective. And because Duchenne is a chronic disease, lifelong treatment will be required. So we also want to see whether there's long-term safety. So all these steps are important, and we have to go through all these steps, and showing that something works in cells is something that we can celebrate, but we have to be aware that the next steps are always more difficult. So if something works in cells, it can still not work in mice. It's possible that maybe it's really easy to deliver it in the cells, but in the mice, it's just not possible to get it to the skeletal muscle. Or it may work in a mouse, but then it may not be safe in the humans. So the next step is always more complicated, and it's always possible that after the next step, you have to conclude that this therapy will not work in patients. So that is why many things will fail along the line, because each step you need to show that it works, and each step is more complex than the previous step. Now, going into the different therapeutic approaches, I'll talk about dystrophin restoring approaches, cell therapy, focusing mainly on exon skipping and AAV gene therapy, and briefly something about gene editing, and then also the two approved approaches that target pathology, vamorolone and givinostat. So starting with cell therapy, and I can be very brief about cell therapy, because for now, there is no evidence that cell therapy works. And this may be counterintuitive because the hypothesis, the theory sounds so appealing. So Duchenne patients don't have dystrophin and have chronic muscle damage. So what if from a healthy individual we isolate muscle stem cells or other stem cells, we expand them in the lab, and then we transplant them in the patients. What they can then do, they can repair the damage, and because they're from a healthy individual, they can also express dystrophin. So we work on two different levels. So that is the theory, and it sounds really appealing, but sadly, the reality is that it doesn't work. And the reason it doesn't work is multiple. First, the stem cells, if you inject them in the bloodstream, will not go to muscle. This is very, very inefficient. So you cannot expect one stem cell that happens to enter a muscle to undo all the damage that is there, and that one stem cell to then produce enough dystrophin to supply the whole muscle. That is not realistic. Even if you inject them locally within the muscle, they will not migrate. They will stay where they are. So again, that makes it very difficult. So the efficiency is extremely low. And also the abundance of the muscle is a problem, because you really need a lot of stem cell to counteract the ongoing pathology. And that is a challenge. So there's currently no evidence that stem cell treatment is effective or that it is safe. If you want stem cell treatment, you can get it because there's people offering it and claiming all sorts of things without any true evidence. If you want to do it, I would strongly argue against it, because you do not know what they will give you. It might not be even stem cells. And if it is stem cells, well, you don't know where they got them from and what it will do. And even if it does no harm, we know there's no evidence that it will do anything therapeutic. It will definitely do harm to your wallet, because the people offering these stem cells as a cure for Duchenne and whatever disease, they charge a lot of money for this. So I would say, don't do this. There is one alternative approach that is currently in clinical development, and that is used by Capricor. And here they don't use the stem cells to restore dystrophin, but they use stem cells because stem cells produce growth factors, and these growth factors can have a positive effect on the muscle repair. So that means that you have this effect as long as the stem cells are in the circulation. And with time, they will disappear because they don't move to muscle, they just circulate, and then they will die. So their approach, you need to treat every 3 months to get new stem cells and to benefit from new growth factors. This approach is currently in clinical development. Capricor filed with the Food and Drug Administration in the US for approval, and the US for now has said that they need more information, because with the current information, they cannot conclude that this is therapeutic. So we'll have to wait and see how this develops. But bear in mind that this is not the traditional stem cell therapy, but a different form of stem cell therapy. Now, moving to exon skipping. So what exon skipping tries to do is it tries to restore the genetic code. And I already explained that in Duchenne patients, the code is disrupted. So we're back to our example of 47 not fitting to 51. But it would fit to 52 because we've seen that that is happening in a Becker patient. So now we're zooming in this process, the splicing process, where all the exons are linked together. And what we would like in a patient with Duchenne, in this case, would be that the cell somehow doesn't include exon 51. Because if it doesn't link 47 to 51, but if it links 47 to 52, then the gene code would be restored and the code would be readable because 47 fits to 52. Now, how can we induce the cell to not include exon 51? We can do this with AONs, antisense oligonucleotides, small pieces of modified RNA that bind specifically to an exon, in this case, exon 51, hide it from the splicing machinery, and now 47 is linked to 52, the code is restored, and a Duchenne patient can make a Becker-like protein. So that is the theory. So where are we now? In the US, there's 4 AONs approved, and they're all of the same chemical modification, the morpholino modification. They're not approved in Europe. They target 3 different exons, so this is a mutation-specific approach. So I use the example of exon 51 skipping because that applies to the largest group of patients, but other patients have other mutations. So they may need 53 skipping, 45 skipping, or skipping another exon. So there's 4 approved AONs targeting 51, 53, 2 of them, and 45. The approval is based only on the restoration of dystrophin. So we don't know yet whether this slows down disease progression. And I would normally say some dystrophin is better than no dystrophin, but the dystrophin levels that we see in these trials usually are around 1% of normal. And then the question is, will that be enough to significantly slow down disease progression? And that is currently tested in clinical trials. So we don't know yet, and this is also why in Europe these compounds are not yet approved. What is clear is that there's room for improvement, because even if 1% of dystrophin will slow down disease progression, more percent of dystrophin restoration, of course, is likely to have a larger therapeutic effect. So there's currently many different clinical trials ongoing, and they try to improve the applicability to go beyond exon 45, 51, and 53 to improve the delivery. So why is there only 1% dystrophin restoration? Because the majority of these AONs does not go to the muscle, but goes into the urine. And to improve the regimen. The current treatment is weekly intravenous infusion. And of course, if you can do it every other week, every month, or even every 6 weeks, that is much less of a burden. So before we go into an overview of the different trials, if you follow this, you will see that when there's results, usually companies will mention dystrophin levels. There's several things that you have to bear in mind here. First is that most patients make dystrophin before treatment. And then the goal is to make more dystrophin after the treatment. So that means you need 2 biopsies. You need a biopsy at baseline and you need the biopsy after the treatment. And then you need to measure an increase. Furthermore, companies will use different methods. They will use different normalizations. So that means that it's really difficult to compare between companies. So if Sarepta is developing 2 compounds and they say we see 1% dystrophin with this one and we see 1% dystrophin with that one, we can compare that because it's the same company using the same method. But if different companies do it, well, we don't know how this compares. So that makes things difficult. And also we know there's variation in how much dystrophin to expect depending on when you take the biopsy. If you take the biopsy later in the trial, probably there's more time for the dystrophin to accumulate. So the point I'm trying to make is that when you see a dystrophin level, it's not automatically that if one company says we have 4% and another company says we have 4%, that they both have equally well-performing AONs. And another thing is that if one has 10% and the other 5%, not necessarily is then their approach better, because there's all these variations. And then finally, we know now that exon 44 skipping is easier. So if we skip exon 44, you will always get more dystrophin than if you skip any other exon. So now going into the different approaches that are ongoing to improve the effects. First, companies that have AONs approved by the FDA, need to do confirmatory studies to show that there is a slower disease progression. And these studies on the next slide, I will show in black. So these are PMO chemistries. And they're also trying to use PMO for additional exons, also in black. Then we have chemical modifications that try to improve the efficiency. And these are new chemical modifications. So I will show these one in red. And because they're new, like, of course, they work better because that's why they were modified, but they're new. So they were not yet tested in humans. So what about safety? It's possible to improve the delivery in general, and these I will show in blue. And the usual way to do this is to link an arginine-rich short peptide to the morpholino. And this works really well in mouse, but it has a very limited therapeutic window, which means that the difference between when these compounds become toxic and when they become effective is very close together. What comes first in mouse is that they become effective before they become toxic. But the question is whether that's also the case in humans. And then finally, what you can try is to improve delivery specifically to skeletal muscle and heart. And these compounds are shown in green. And this is done by linking the compounds to exon skipping compounds to antibodies to transferrin receptor 1. So transferrin receptor 1 is an iron transporter that's expressed on all cells in the body, but more on muscle and heart cells. So we can use it to enrich the delivery to skeletal muscle and heart. But these antibodies are much bigger than these short peptides. So it's possible that the body will respond to this and starts making antibodies to the antibodies. And of course, these antibodies may also interfere with iron transport, which we need. So this is the overview of currently ongoing or recently stopped clinical trials. And you will see there are now trials for 5 different exons ongoing. For exon 51, it's very crowded. So there's 6 trials that were ongoing. So the 2 with the arginine-rich peptides were stopped. And they were stopped either because they were not efficient. So that's this one, the one from PepGen, or because of safety concerns. That's the Vesleteplirsen from Sarepta. So as I said, if you develop new things, it might not be safe in humans. And that is sadly what happened here. So they had to stop it. The Eteplirsen confirmatory trial is still ongoing. And then there's also new chemical modifications from BioMarin and Sky Therapeutics, and this muscle-specific delivery developed by Dyne. For exon 45, there's 2 trials ongoing. One, the PMO, the approved casimersen, where the company has to show that indeed there's a slower disease progression, and one with an arginine-rich peptide. This is a slightly different peptide from the one where the trials are stopped. It's a cyclic peptide and it's less arginine-rich. So the hope is that this will have a better safety window. But the trial just started, so we need to wait and see. Then for exon, and this is developed by Entrada. For exon 53, there's also currently 3 trials ongoing, confirmatory studies for golodirsen from Sarepta and viltolarsen from NS Pharma. For viltolarsen, after 1 year, the primary endpoint was not met. And well, I'm not sure whether that means that the treatment doesn't work. I think it means that if it works, we need a longer time to show whether it slows down disease progression than 1 year. And WAVE is also doing a clinical trial here with a new chemical modified oligo. For exon 44, there's 3 trials ongoing. There's one with a regular PMO from NS Pharma. Entrada is also focusing on exon 44 with a cyclic peptide that aims to improve delivery in general. And Avidity is doing a clinical trial with a transferrin antibody receptor mediated delivery, so muscle-specific delivery. And then finally, there's 2 trials for exon 50 targeting. Again, one from Entrada with this cyclic delivery peptide that is planned and hopefully soon will start. And NS Pharma has one ongoing with a regular morpholino to skip exon 50. So there is a lot happening here. But of course, the challenge is currently we have no evidence that there's a slower disease progression. This needs to be shown. And there's all these new approaches that so far may look promising, but as you see, also with new approaches, we don't know what the safety will be in patients. And then sometimes this means that we have to stop the clinical development because things are not safe or because they do not work out. Now, moving to gene therapy, because also a lot is happening here. So what is the premise of gene therapy? Duchenne patients cannot make dystrophin because their gene code is unreadable. What if we make a functional copy of the gene with a readable code and we give it to muscle? Now, how do you deliver genes? We're not very good at this as humans, but there is a virus, the adeno-associated virus, that's actually very good at delivering things to skeletal muscle. So this means you can only use this if you didn't see the AAV virus in the past, because if you did, there will be immunity and there will be an immune response. So this limits things because about 50% of patients will have encountered AAV in the past. AAV is also small, and that means only small genes fit. And sadly, the dystrophin gene happens to be one of the largest genes we have. So we had to engineer, we had to make a microdystrophin in order for it to fit. But of course, if it's a microdystrophin and it's only 32% of the normal dystrophin, then we know it's not fully functional. It will be partially functional, and it's significantly shorter than the shortest Becker dystrophin that we have. So we need in the gene, in the viral microdystrophin gene, we need the code for this microdystrophin, also a volume switch to make sure it's only expressed in skeletal muscle and heart, and some viral sequences because it needs to go into the AAV capsids. So I mentioned already, we need to engineer, we need to have a minimalistic approach, and different companies have different ways on how they made their microdystrophin. So what we see this first click domain, the actin binding domain, is present in all of them. The second click domain, the cysteine-rich domain that connects to the connective tissue, is present in all of them. But then what differs is how much or which parts of this connecting domain they have. So they have either 2 or 3 of the hinges, and they have either 4 or 5 of the repeat spectrin domains. So it varies. If you ask any company, they will all say ours is the best, but you can see there's more similarities than differences, and they're all significantly shorter than the full-length dystrophin and even the Becker dystrophin. So I mentioned already that there's some considerations with AAV because normally AAV doesn't cause a disease. So 50% of us will have AAV antibodies, and we didn't realize we did because we never realized we were sick. However, when you encounter AAV, normally you will have a few viruses and they will start to expand, but it will not be that many. If you use AAV for gene therapy, the doses are much, much higher. So they're about 2,000 times the number of erythrocytes, so red blood cells that we have in our body. So they're really, really high. And of course, if you use these very high doses, this will cause side effects. So the liver will get very high exposure because, well, we say AAV goes to muscle and heart, but the liver will get the majority of viruses. So that means side effects in the liver. And also, if you use this high of a dose, there will be an immune response, an innate immune response, or an immediate response, and an adaptive response. So this will cause side effects. And also, once you have been treated with gene therapy, you cannot retreat because there will now be antibodies to AAV. So where are we now for gene therapy? There have been clinical trials done by different companies, and well, Elevidys from Sarepta is approved. We'll get to that. But what we know now is patients were treated without antibodies, and we have probably over 1,000 different patients treated either in a clinical trial or in a commercial setting. What we know from that is that it works. If you treat patients with this AAV microdystrophin gene therapy, the muscles will start to produce microdystrophin. But we also know that there are side effects, and these can be severe to very severe. And so far, 5 patients have died after receiving AAV gene therapy, 3 in a trial from Pfizer and 2 after receiving Elevidys. The side effects or the risk of severe or very severe side effects are related to the dose. But the thing is that we cannot really say, well, then we give a lower dose, because if we give a lower dose, the microdystrophin levels will be much, much lower and probably not be therapeutic. We also know that the older the patients are, the more likely it is that they get severe side effects. And this is because, well, when you're older, you're usually heavier, so you will get more viruses because you get X viruses per kilo. But also, if you're older, you will be in a worse physical condition. So your ability to cope with these very severe side effects will also be less because there is diminished heart function and diminished respiratory function already. So I mentioned already these acute side effects that can be as bad even as death, but also liver damage has been reported, transient kidney failure with patients needing temporary dialysis. And all of this is due to an immediate immune response to AAV. But it's also possible in some patients that there was a later response. So they say coped with the first few weeks of side effects, and then later, after 3 months or even later, suddenly they had muscle breakdown and inflammation of the heart. And this turned out to be due to an immune response to the microdystrophin. And it was not seen in all patients, in only a few patients. And then we can ask ourselves, why is that? Because the immune system recognizes foreign proteins. Duchenne patients don't make dystrophin, so shouldn't the microdystrophin trigger an immune response in all patients? Now, I'll try to explain why that's not the case. If we have the full length, the normal dystrophin here and the microdystrophin here, what we know is that almost all patients are able to make a very short form that's mainly expressed in the brain that contains only the ends. So this last piece. So that will be there in more than 99% of the patients. So this part of the microdystrophin, this last domain connecting to the connective tissue, is there. So this is not new. Now, most patients also will have a mutation in the middle of the gene here. So that means they make the beginning. The immune system is known to this first part of the protein. It's not functional, but it will have encountered it. So now if we have a microdystrophin having this beginning that has been seen by the protein or by the immune system, and this last domain that's also been seen by the immune system, this is not new. Patients who have a deletion at the beginning of the gene, however, they can never make this first part connecting to the skeleton of the muscle fiber. They cannot make it. So now when they get a microdystrophin, they will start to express protein domains that the immune system has never seen. And then the immune system will recognize this as a foreign invader, and it will start to attack all the cells that express this, which will be the muscle and the heart tissue. And of course, then you're worse off than if you have dystrophin, if you have Duchenne, because now you start to eliminate muscle and heart cells even more rapidly. So for that reason, patients who have deletions at the beginning of the gene are now excluded from trials or receiving microdystrophin commercially. So functional effects that we have seen so far, we know there's microdystrophin, as I mentioned already. And when this was tested in open label phases, we could see that the function was better than the natural history. But there was a but, and that is that with the gene therapy, because you inject such a high dose of AAV viruses, patients are treated with a very high dose of steroids for a few months. And we know now that this will also slow down disease progression. So that means that the improvement that we saw in these patients might not be due to the microdystrophin, it might be due to this higher dose of steroids. And of course, the challenge with microdystrophin is we don't expect this to improve patients. What we expect is it to slow down the disease trajectory. And what we've currently seen is no evidence of a slower disease progression. Does that mean that the treatment doesn't work? Well, not really, because, well, there was a trial with the placebo group, but the suboptimal dose was given, so maybe that's why we didn't see it. And then there were 2 clinical trials, but they were done in patients who were stable. And if you want to show that something slows down disease trajectory, you will not be able to measure this in stable patients. So sadly, we don't know whether the treatment works or not, and if it works, to what extent. And there are additional studies, but they're still ongoing or on hold. Because back to the Elevidys, I mentioned already, Elevidys is approved in the US. It's not approved in Europe. So it's been evaluated by the EMA, and the Committee for Human Medicinal Product gave a negative opinion last July. it's approved in the US based on its ability to restore microdystrophin. The confirmatory studies didn't show that there was a slower disease progression, and still it was approved. There was also accelerated approval in non-ambulant patients, and here confirmatory studies still need to be done. But because sadly two non-ambulant patients died after receiving AAV microdystrophin gene therapy, Elevidys, these trials are currently on hold, and the company has to investigate what was the cause of these patients. So they both had liver failure, but we need to understand more to see whether everyone is at risk or only subgroups, etc. So for ambulatory patients, Elevidys treatment in the US is still there, and these trials are ongoing. But for non-ambulatory patients, the treatment has stopped and the trials are on hold. And patients should not have a deletion at the beginning of the gene, and this is because these patients are at risk of having an autoimmune response. So there's still some challenges towards the future. We don't know how functional these microdystrophins are. We know they're functional in mouse and in human or in dogs, but mice and dogs walk on 4 legs while humans walk on 2 legs. So the dynamics are different. So we don't know yet, and as I mentioned, also from the clinical trials, we don't know yet whether microdystrophin treatment will slow down disease trajectory. What we do know is microdystrophin is not fully functional. And that means that with time, there will still be muscle damage and there will be muscle turnover. So with time, the gene copy of the microdystrophin will be lost. And that therefore, with time, the microdystrophin levels will go down. What we don't know is how long this will take. Does it take 2 years? Does it take 20 years? Of course, that's a big difference. We do know that there will be an immune response to AAV, so we cannot treat patients with pre-existing antibodies. We cannot retreat patients when the microdystrophin is lost. Sarepta is currently doing clinical trials to see whether we can treat patients with pre-existing antibodies by targeted immune suppression. So these clinical trials are ongoing. And finally, we know that some patients will have an immune response to microdystrophin, and these are for now excluded from receiving the gene therapy. So towards the future, we need to monitor patients, because for now, we have no evidence that there are any functional effects. And of course, also for the long term, we don't know what the side effects will be. So patients need to be monitored. There are studies to see whether we can deliver larger dystrophins with the Indian system. This is currently only tested in mice, and it requires a double dose of AAV, so that will be challenging for now. There's companies focusing on trying to improve AAV. So can we make an AAV that goes better to the muscle, so less to the liver? And those are called myo-AAV, and Solid is currently doing a trial with the myo-AAV. Companies are developing different methods to deliver, but this is in an early stage. And we're trying to retreat patients. And I mentioned already that Sarepta is doing a trial to see whether patients can be treated if they have pre-existing immunity. And also there's research ongoing to try and reduce the immunogenicity of the microdystrophin so that also these patients with the deletion at the beginning of the gene can be treated. Now, very briefly going into gene editing, because it combines aspects of the microdystrophin, so the gene therapy and the exon skipping. So what is gene editing? You probably heard of it, CRISPR-Cas9. It's basically a scissor that cuts in the DNA and a GPS system that tells us where to cut. So why would we want to do this? Well, with exon skipping, you create a Becker-like dystrophin. So it's much larger than with microdystrophin and therefore likely more functional. But you need repeated treatment. And currently the approved exon skipping compounds, you need to do weekly intravenous infusion. Because you target the RNA, and the RNA will be degraded, and then you need to repeat the treatment. What if we could make the exon invisible on DNA level? Then you need to treat only once, because every time a copy is made on RNA level, this will be a restored copy. So how can we do that? We can do that by, for instance, cutting the beginning of exon 51. the repair system will try to repair it, but it will not do it properly. And then exon 51 will now be hidden. So that means for this patient with the deletion of exon 52, you see 51 doesn't fit to 53, but when exon 51 is hidden, 50 will fit to 53. So now we get an RNA that allows a Becker type dystrophin. And you have to do this only once because it happens on DNA level. This is early stage yet, so it's tested in cell models and in animal models, but not yet in clinical trials in Europe or the US. And also here it's important to bear in mind the expected treatment effect depends on the time of intervention. So you can think, well, because it's on DNA level, it should make everything better. But if in a very dystrophic muscle, you now correct the fibrotic tissue, you correct on DNA level the lack of dystrophin, that doesn't mean that this fibrotic tissue will become muscle. It's fibrotic tissue, it will not express muscle. Even though the DNA is there, it will not express dystrophin and it will not turn back into muscle. So again, also here, you need to treat the majority of muscle fibers in all the muscles, and it will not restore a normal dystrophin, like all the other approaches that I discussed. So how to improve? Also here, you need to deliver 2 AAVs, one for the scissor, one for the guide. So we need to improve the delivery with AAV because with the current approaches, we cannot double the dose because that would lead to severe side effects. So there's that safety concern of the high dose of AAV, but also the wrong edits because you do this on DNA level, and that means you cut and you cannot undo. And if it cuts also somewhere else where you didn't want it to cut, and suddenly you now have activated a gene that's involved in cancer, you have a problem. And then there's the risk of immune response. So AAV, which is the delivery tool, that will induce an immune response. But also the Cas9, so the scissor protein, that comes from bacteria. So it's possible that cells that express this scissor will now be seen by the immune system as cells that have been infected by a bacterium, and then they will be targeted. So that's another autoimmune risk. And these things are all still need to be addressed. So finally, moving on to approaches targeting pathology, and these are both the vamorolone and givinostat. So starting with vamorolone, which is a steroid-like compound. And steroids are the standards of care, and they do many things, and probably the main way they act is that they inhibit inflammation and then protect against muscle damage. So that slows down pathology. It doesn't address the lack of dystrophin, but it slows down all the pathological processes. Glucocorticosteroid like prednisone and deflazacort also bind the mineralocorticoid receptor. And they induce a high blood pressure, which is not a good side effect because Duchenne patients have cardiac problems, and then high blood pressure is counterproductive. Also, if you give external steroids, the adrenal glands that normally produce steroids and cortisol stop working because, well, they don't need to work. The compounds are delivered. And that means that if something goes wrong, like you have an accident or a fracture, you need stress dosing because normally when we are in stress, we will start producing cortisol, but the adrenal glands of patients who are chronically treated with steroids, they don't do this. And also this causes side effects, this adrenal suppression. And finally, steroids can move to the nucleus of the cell and activate the transcription of all sorts of genes that we don't want, and these also cause side effects. And I'm sure that you're all familiar with the many side effects that chronic steroid use involves. So the idea then is, can we make steroids that have less side effects? So we have prednisone here, deflazacort here, so you see they are very similar. And here we have vamorolone. So vamorolone is very similar to prednisone, but due to these minor chemical changes, vamorolone is not able to go into the nucleus. So we expect less side effects. We also know that vamorolone will cause a lower blood pressure. So that's a positive thing. It still inhibits inflammation, so that's good. And also it still suppresses adrenal glands, so that is not good. So it will not negate all side effects, but the hope is that it negates some. So what did it do in clinical trials? It has been tested in patients 4 and older, and it showed that it's similar in efficacy to daily prednisone. early on, it showed less side effects than prednisone, with no signs of osteoporosis or growth stunting. But the weight gain, that's one of the big side effects, still happened in vamorolone. And I say early on because it's a new developed therapy. So what we don't know is what will happen if patients are treated with this for 5 years, 10 years. That data is still not out there. But based on the trials and the fact that it has a similar efficacy as prednisone, and for now seems to have less side effects, this compound is approved by the EU, in the UK, by the MHRA, and in the US. And the long-term effects are not known, as I said. And well, it's approved by the European Medicine Agency, but that means that country by country, it now needs to be marketed. So it's not yet marketed in different countries. I don't know whether it's marketed in Slovenia. I know it's not yet marketed in the Netherlands. And usually this can take 2, 3, 4, 5 years even. Finally, HDAC inhibition, givinostat. So this is a complex approach, and what I can tell you is that I try to explain things in simple terms, and then when I published that text on the World Duchenne Organization website, the company developing givinostat, Italfarmaco, said, "Well, if we give you a sponsorship, would you like to work with graphic designers to convert this into a cartoon?" And I said, well, yes, that's of course very nice. So they sponsored this. So Italfarmaco, who developed givinostat, sponsored making the cartoon, but they were not involved in how the cartoon was and what it said. So if you want to read the cartoon, there's a QR code that you can scan, and this will bring you to the cartoon. So the text is mine, the graphic design is by the people who made the cartoon, and the funding was by Italfarmaco. So for full disclosure. So I'll use the pictures of this cartoon to try and explain how givinostat works. And first, what we need to understand is how normal muscle repair works, because also in healthy individuals, sometimes the muscle is damaged. And then step one is that the immune cells will clear the debris. And then the fibroadipogenic cells, the FAP cells, they will make the structure, they will make the connective tissue, and then finally the muscle repair will happen. So the muscle stem cells will start making the muscle. So imagine or compare it to a kitchen. So you had an explosion in your kitchen. You first need to clear away all the debris. That's what the immune cells do. Then you have to restructure the walls. That's what the FAPs do. And then you put in your kitchen appliances and your tools and your utensils. That's what the muscle tissue repair, so the muscle stem cells do. Now, what goes wrong in Duchenne patients is that there's continuous muscle damage. So there's chronic inflammation. Because there's chronic inflammation, there's too much damage. So the immune cells, they clear away the debris, but they also start to damage the muscle. So the part of the kitchen that's actually not damaged is also now damaged because the immune cells are overactive. The FAP cells that normally should make the connective tissue, so they should repair the walls, they are too active. They make too much connective tissue, so the walls become way too fat, and of course that means there's much less room for you to build your kitchen. And the same in the muscle, there's less room for the muscle to be because there's too much connective tissue. And then finally, the muscle stem cells get the wrong signals. So instead of starting to repair, the FAP cells say, well, maybe you should also convert yourself into a FAP cell and start to contribute to the formation of connective tissue. So these things, due to the chronic inflammation, these things go wrong. And one enzyme that plays a role in this is the histone deacetylases. So acetylation, so you have acetyl group that you can add to protein, and the cell uses this to influence the protein activity. So you can remove an acetyl group, and then usually the protein will be in turbo mode, or you can add it, and then it will be in eco mode. And you have enzymes that add or remove these modifications. Well, the enzymes that remove the acetyl groups, the histone deacetylases, the HDACs, are overactive in Duchenne. And this makes the pathology worse, because now you remove it, and well, turbo mode sounds nice if you're in the car, but you don't want to constantly go 100 kilometers per hour. There are many areas where you don't want that, where you want to be able to go slower. So now these enzymes are in a perpetual turbo state, and that makes things work. So this inflammatory response is too active, and it starts to produce extra damage. The production of fibrosis is stimulated too much, which again is not a good thing. And this then suppresses the regeneration. Now, if these pathological processes are worsened because HDAC enzymes are too active, then you can of course think, well, if that is the case, maybe we should try to inhibit these overactive enzymes. And that's what you can do with givinostat. So givinostat is an HDAC inhibitor, so it inhibits the activity of enzymes that remove these acetyl groups. So what would you expect if you inhibit HDAC? You expect that the chronic inflammation will be better, so there will be less damage, and there will not be that active an inflammation. You expect the Fab cells, the fibroadipogenic stem cells, you expect them to go back to their supportive role to make connective tissue, but not too much, and to stimulate the muscle stem cells, to repair the muscle. So that is the theory. Then, of course, we ask what happened in reality. So there have been clinical trials for givinostat, and in the first trial, they took a biopsy at baseline and after 1 year of treatment, and there was less fibrosis after 1 year of treatment. There was also a placebo-controlled trial where the patients who were treated had a slower disease progression. So both groups, the placebo group and the treated group, they both worsened compared to baseline, but the patients who were treated worsened less. And this was also seen on secondary endpoints. So the primary endpoint was how quick patients could walk up 4 stairs, but also there were other outcomes like the North Star and MRI fat filtration, where treated patients did better. So the FDA has approved this treatment for patients 6 years and older. The EMA approved this treatment for ambulant patients age 6 and older, and the approval was recently, and so it's conditional. So also here, like with the vamorolone, the marketing will happen on a country-by-country basis. So towards the future, what we know that with dystrophin restoring approaches, we can restore dystrophin, but for none of these dystrophin restoring treatments do we have evidence that it slows down disease progression. For the approaches that target pathology, for vamorolone and givinostat, this evidence is there. We know it slows down disease progression, but of course it doesn't bring back dystrophin. In the future, what is likely is that we need a combination, because the approaches that restore dystrophin restore partially functional dystrophin. So pathology will still happen. So we need also to address the pathology. So this towards the future. Well, thank you very much for your attention. If you have any questions, then feel free to email me. My email address is here. And also, if you want to know more about this and other compounds, there is an overview that I generate on the Treat&D website, where I describe the different types of therapies that are in clinical development for Duchenne and the current state. And it was last updated mid-August, so it's still very recent. So thank you very much for your attention. And by all means, feel free to email me if you have any questions.